So let's talk about pharmacodynamics of peptides. Pharmacodynamics describes what a molecule does to the body. Uh, some key pharmacodynamic characteristics of peptides include that their biological effects occur through receptor binding, as I mentioned, triggering intracellular cascades, cellular function response, and then further signaling out to other cells. What's important to understand here is that once this occurs, the peptide no longer has to be present at the receptor on the cell surface for these cascades to be ongoing in the downstream biological effects. They can literally persist for hours or even days after the peptide is no longer present at the receptor or even present in the plasma. So, keeping this in mind, when we talk about plasma half-lives of peptides, it can differ how that affects peptides in comparison with more traditional drugs. Plasma half-life is a very useful way that we try to establish the biological activity duration of more traditional small molecule drugs, but it doesn't apply so well to peptides. Again, because these downstream effects can keep going much long after the peptide is actually present. So what's important to understand here is that there might not be an objective lab parameter that you can actually pull to measure how long a peptide is actually working. And the effects need to be assessed on the functional response scene as opposed to pulling a lab to see how long and how well it might be working.